ഹലോ ഫ്രണ്ട്സ്, എല്ലാ കൂട്ടുകാർക്കും പുതിയൊരു വീഡിയോയിലേക്ക് സ്വാഗതം. ഇന്ന് നമ്മൾ ഡിസ്കസ് ചെയ്യുന്നത് സ്റ്റേറ്റ് എലിജിബിലിറ്റി ടെസ്റ്റ് ജാനുവരി 2022 പ്രീവിയസ് ഇയർ ക്വസ്റ്റ്യൻ പേപ്പർ അനാലിസിസ് പാർട്ട് 3. Question number 10. In urea cycle, the hydrolysis of L-arginine to L-ornithine and urea is catalyzed by option A arginase, option B fumarate, option C glutamate, option D phosphate synthetase. In urea cycle, the liver converts ammonia to less toxic urea. Urea so formed is transported to kidneys and get excreted in urine. The process of production of urea is known as urea cycle. It was discovered by Hans Krupp and Kurt Hanslitt in 1932. Hence, it is also known as Krupp-Hanslitt cycle. Urea has two amino groups in its structure, one derived from ammonia and the other from aspartate. Urea cycle includes 5 steps involving 5 enzymes: synthesis of carbamoyl phosphate, condensation of NH4 plus ions and carbon dioxide. In presence of enzyme carbamoyl phosphate synthetase forms carbamoyl phosphate. The formation of carbamoyl phosphate requires energy. This step consumes 2 ATPs. Formation of citrulline. Citrulline is synthesized from carbamoyl phosphate and ornithine with the help of enzyme trans-carbamylase. Synthesis of arginosuccinate. The enzyme arginosuccinate synthase condenses citrulline and aspartate to form arginosuccinate. This step requires ATP. Cleavage of arginosuccinate. Arginosuccinate lyase cleaves arginosuccinate into arginine and fumarate. Formation of urea. Arginase is the fifth and final enzyme that cleaves arginine to form urea and ornithine. Ornithine enters the mitochondria where it is reused in the formation of citrulline. Once again, formation of urea. Arginase is the fifth and final enzyme that cleaves arginine to form urea and ornithine. Ornithine enters the mitochondria where it is reused the formation of citrulline. So the answer is option A, arginase. Question number 11. The basic principle of this instrument is sedimentation. Option A, chromatography tube. Option B, centrifuge. Option C, photometer. Option D, spectroscopy. The centrifugation based on the principle of sedimentation. The acceleration of centripetal force causes separation of denser particles along the radial direction at the bottom, and lighter particles will move to the top of the tube. The particles have higher density settles at bottom, that is sediment, and lighter float to the top. The concept of centrifugal force is applied in rotating devices. Centrifugal force is the force that draws a rotating body away from the center of rotation. So the answer is option B, centrifuge. ഓരോ ഓപ്ഷൻസും കൂടി നമുക്കൊന്ന് അനലൈസ് ചെയ്ത് പോകാം. Chromatography. Chromatography is an analytical technique commonly used for separating a mixture of chemical substances into its individual components so that the individual components can be thoroughly analyzed. There are many types of chromatography. Example, liquid chromatography, gas chromatography, ion exchange chromatography, affinity chromatography. അടുത്തത് photometer. The device which is used to measure the candle power or luminous intensity of a given source of light is known as photometer. അടുത്തത് spectroscopy. Spectroscopy is mainly used to identify and elucidate the elements and compounds of atoms and molecules. They are measured by examining the absorbed or emitted radiant energy by the sample or object. Here, the beam of electromagnetic radiation, like infrared, UV rays, etc., are passed on the sample, and the response of the sample is measured through the wavelength of the electromagnetic spectrum applied from the external energy source. Question number 12. Type of speciation caused to reproductive isolation. Option A, allopatric. Option B, sympatric. Option C, Patrick. Option D, none of the above. The biological species concept defines a species as a group of individuals living in one or more populations that can potentially interbreed to produce healthy, fertile offspring. Other species concepts exist and may be more useful for certain types of organisms. Species are kept distinct from one another by pre-zygotic and post-zygotic barriers. These barriers keep organisms of different species from mating to produce fertile offspring, acting before and after the formation of a zygote respectively. These barriers maintain the reproductive isolation of species. New species formed by speciation, in which an ancestral population splits into two or more genetically distinct descendant populations. Speciation involves reproductive isolation of groups within the original population and accumulation of genetic differences between the two groups. In allopatric speciation, groups become reproductively isolated and diverge due to geographical barrier. In sympatric speciation, reproductive isolation and divergence occur without geographical barriers, for example by polyploidy. So the answer is option B, sympatric speciation. ഓക്കെ ഫ്രണ്ട്സ്, വീഡിയോ നിങ്ങൾക്ക് ഇഷ്ടമായ ലൈക്ക് ചെയ്യുക, ഷെയർ ചെയ്യുക, സബ്സ്ക്രൈബ് ചെയ്യുക, ബെല്ലൈക്കൺ ഒന്ന് പ്രസ് ചെയ്യുക. താങ്ക്